Well, the question that we used was, how does temperature affect the rate of the enzyme activity? Our hypothesis was, if the temperature is increased, the rate of the reaction of the enzyme, which is liver, may increase. However, according to our notes, it says that it will only increase to a certain point before the enzyme denatures. If the temperature decreased, the rate of the reaction of the enzyme, which is the liver, will increase. The materials that we used during this lab were 1 hot plate, 3 test tubes, 3 beakers, 4 cylinders, a stirring rod, goggles, aprons, knife, cutting board, crushed ice, thermometers, beaker tongs, crushed liver, 10 millimeters of 3% H2O2. First, we took 3 beakers and 3 test tubes. We filled 1 beaker up with water and placed it on a hot plate to create a hot water bath. Then we put 1 gram of crushed liver in each test tube. We placed 1 test tube in hot water bath on the hot plate. We waited until the temperature turned to 45 degrees Celsius. Then we placed the liver from the hot test tube into the first cylinder containing the 10 milliliter of 3% H2O2. We did the same thing with the second test tube, except we waited until the temperature turned 60 degrees. We placed the third test tube into a beaker with crushed ice and waited until the temperature reaches 0 degrees Celsius. Then we placed the liver in from the cold test tube into the second container, which is a cylinder, containing 10 milliliters of 3% H2O2. Then we put the leftover crushed liver directly into the third container, which contained H2O2 solution. Then we observed what changes were happening and how much the oxygen bubbles were produced in each cylinder, and we recorded the data and observations into our notebook. For the hottest temperature of liver, which was 60 degrees, it produced 5 milliliters of gas. For the hot temperature, which was 45 degrees, it produced 7 milliliters of O2. For the controlled liver at room temperature, produced 12 milliliters of O2. For the cold temperature, which was 0 degrees, it produced 13 milliliters of O2, which is weird because we expected the normal one to be produce the most, but apparently maybe it's because the reaction was slow, but a higher O2 was produced. From our own explanation and understanding, we can explain that the cold temperature slowed the enzymes and substrates, which caused a prolongation of the bonding rate of the creation of the enzyme substrate, which is why it took really long for more O2 to be produced.